Welcome to today's refresher lecture. I'm Dr. Leslie Blankenship-Williams, and today we are going to review the concepts of nucleic acids. You may recall that there are actually 4 different classes of biological molecules. There are carbohydrates, lipids, proteins, and nucleic acids. Today, we're going to be looking at the structure and function of nucleic acids with an emphasis on DNA. With any biological molecule, you have building blocks, and the building blocks of nucleic acids are nucleotides, so we'll start there. From there, we'll build up and look at the structure of DNA. And then finally, we will take a look at what do we do with DNA. One of the things that we need to do with DNA is, of course, copy DNA to make more DNA. So we'll look at semi-conservative DNA replication, and of course, the whole purpose of DNA is to code for your proteins. So we'll look at gene expression, or the process of going from DNA to a protein. Okay, starting with a nucleotide. When you take a look at a nucleotide, which is the building block for all nucleic acids, the nucleotide itself is actually broken up into 3 components, which is a little unusual for a monomer. One of the things that I want to draw your attention to is this sugar down here. This sugar is going to be a component of every nucleotide. Then we move up to the phosphate group. And then finally, we move up to the nitrogenous base area here. Thus, every nucleotide has those 3 components: sugars, phosphate groups, and nitrogenous bases. When you take a look at a special type of nucleic acid, the sugar and the phosphate group remain constant with every nucleotide. But the nitrogenous base can change. In DNA, there are 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. Now that we've taken a look at the nucleotide structure, let's take a look at how we put them together to make a polynucleotide, which is the polymer form of nucleic acids. If you notice, over on this side, here, we have the nucleotide, and then over here we have 5 nucleotides stacked on top of each other. So what you do is you take the whole nucleotide and you move it over down to the bottom and then start stacking right on top of each other. As that happens, you form covalent bonds between the individual nucleotides to make a long chain of a polynucleotide. So if you look at the structure of that long chain, you'll notice that the phosphates and the sugars comprise what looks to be almost a zigzag shape, and that of course is going to be the backbone. And then you might notice that the nitrogenous bases are sitting out a little bit by themselves. As you look at the nitrogenous bases, you might notice that there are actually, once again, 4 different letters, which comprises the 4 different nitrogenous bases. We have adenine, cytosine, thymine, guanine, and thymine then again at the bottom. And you'll notice each nitrogenous base is denoted by a single letter. So now that we've covered a polynucleotide, let's think about how we're going to put together DNA. DNA is special because DNA is not just one polynucleotide, but actually 2 of those strands put together in a special arrangement called a double helix. So let's take a look at this slide, and we'll start with the center picture. So in looking at the center picture, the area that I'm pointing out right now is actually that single strand. But then notice there's another strand over here too. When we take a close look at the strands, we'll notice that the nitrogenous bases of those 2 strands are facing each other, and one nitrogenous base always matches another nitrogenous base. So let's take a look. Up at the top we have T and A, and then we have G and C. Here again we have C and G, and then finally again we have A and T. We call those the base pairing rules, in that A and T always go together and C and G always go together. A really neat way to remember this is that the curves of the letters go together. So a C is made with a curve, so is a G. So the C's and G's go together. An A is made with a straight line, and so is a T. So the A's and the T's go together. And that's a really clever way to remember who pairs with whom. And those rules are always hard and fast. In other words, if you have a thymine, you know that an adenine is always going to be paired with it. If you have a cytosine, you know a guanine is always going to be paired with it. Another feature of this double helix is that the strands do not run in the same orientation. You might notice one strand seems to be running in one direction, but the other strand is flipped and running in the other direction. We call that an anti-parallel arrangement. Once we have our two strands held together by a hydrogen bond, the chemical structure is actually not straight up and down like a ladder, but rather it rotates some. That rotation creates what we see as the double helix, and that rotation is visible on this slide with both pictures that are located on the outskirts. Okay, now that we've covered the structure, let's go through how we are going to replicate DNA. Anytime you have a cell that has its DNA and you want to make a new cell, you also need to replicate the DNA in that cell, such that the new cell also gets a copy of that DNA. After all, DNA is the genetic code of life. So let's assume that we are going to start with one cell and we're going to break it off into two identical cells. Thus, each cell needs its copy of DNA. So we're going to start with the original cell. We'll call that the original or the old DNA, which is shown here in blue. Once again, notice A pairs with T and C pairs with G. Take a moment to confirm that you notice A is pairing with T and C is pairing with G in this double helix stretch. Okay, so now that we know that, what we're going to do is we're going to take that parental or old model of DNA and we're going to pull it apart such that each rung is exposed. Now, that provides a template for us. If we take a look at one strand, let's start with the one that starts with an A and goes down C, G, A, and T. Take a look at the new nucleotides that are matching it. They, of course, are following the base pair rules, T, G, C, and so forth. What nucleotides would we add after? Well, of course, after T, G, and C, we would add T and A. And then if we look at the other strand, we notice the same base pairing rules take effect. At the end, we end up with two identical strands. Please take a moment to confirm that you notice both of those end strands are identical. In other words, they have the same sequence of letters. Okay, we call this semi-conservative replication. The reason why we say semi-conservative is because half of a new DNA strand is new and half is old. So we denote this by showing that each of the new daughter DNA strands are blue and white, the blue representing the old and the white representing the new. The word semi means partially. Conservative means that you retain. So what we are saying is that half of the DNA is retained when we go through DNA replication and half of it is new, hence the term semi-conservative replication. Here is a picture showing more of this semi-conservative replication. In this case, you can see that the original double helix is being pulled apart, and as each old strand is being exposed, a new strand is being synthesized around it. Once again, take a look at one base pair and you'll see C is always pairing with G and A is always pairing with T. Okay, now that we've covered how DNA is replicated, let's take a look at what we do with DNA when we want to allow it to code for something. It's great that we can replicate our DNA and put it into new cells, but the real function of DNA is really to code for the construction of stuff in your cell. And not just any stuff, but most specifically, proteins. So the process of going from the alphabet of nucleic acids to the alphabet of amino acids, which is the material that makes up the protein, actually is a little bit difficult. So we're going to go through this process. Also of an issue is that you have thousands and thousands of genes in each one of your cells. A gene is going to be a stretch of DNA that codes for one particular product. So you don't necessarily want to turn them all on at the same time. An analogy to this concept is if you take a look at a computer. A computer could have hundreds and hundreds of programs loaded on it. But when you sit down in front of a computer, you don't want all of those programs to be running at the same time because you don't need most of those programs. Instead, you are going to turn on just the one program that you need to work on at that moment. So what do? You take your mouse and you double click on the icon and open that program. And that tells the computer, this is the program we want to access, so let's go ahead and bring it up for this person. And the rest of the programs are left alone. The same thing is true with our DNA. We don't want to access all the DNA at the same time. Instead, we only want to access what we need for the moment. We call that turning on a gene, just like you would turn on a program. So in the process of turning on a gene, you take a segment of that DNA and you say, this is the segment that I want to activate. How do we do it? We go through a process where we make an RNA copy, and from that RNA copy, it goes out of the nucleus and into the cytoplasm, and from there, we translate it into the alphabet of amino acids that makes proteins. Let's take a little bit closer look. So again, gene expression goes from DNA to RNA, and then from RNA to a protein. There are words that make up each one of these processes. When we go from DNA to RNA, we call that transcription. And the reason it's called transcription is because to transcribe essentially means to copy. So when you transcribe DNA, you are making a mobile copy. Interestingly, RNA is also a nucleic acid. And so because it's made of the same alphabet of nucleotides, it's really much just a copy of that DNA. The RNA is mobile though, and so the RNA can go out and become translated into a new alphabet. So when you go from one language to another, we call that translation, and the same thing is true in the cell. The language of nucleic acids is not the same language of proteins, so we need to translate it. So again, the whole process that makes up gene expression is going from DNA to RNA. We call that transcription, or essentially to copy, and from RNA to protein, and that is translation, because we switch alphabets. Taking a closer look at transcription, we'll see that RNA polymerase is the enzyme that is going to make a single strand of RNA. So what we are doing here is we are taking a single strand of RNA that we are copying straight from the DNA. So if you might notice if you look at the pink, which is meant to be the RNA, versus the blue, which is meant to be the DNA, we'll see that the base pair rules still sort of apply. In other words, we still see C's and G's are always paired up. And in some sense, A and T is paired up. But if you see U is a little different. It turns out that RNA doesn't quite use the same alphabet as DNA does. It's a lot like going from American English to British English. It's pretty much the same, but there's a few variations. So we'll see that the RNA is just a little bit different than the DNA is. Regardless, we still work within the same basic framework, and so we still call this transcription. The RNA makes a mobile copy of the DNA, and the RNA is then going to go out and find a ribosome. It is the ribosome that is responsible for translation. In this sense, what happens here is the ribosome finds the small subunit, the RNA finds a small subunit of the ribosome, and then we start the process of grabbing the correct amino acids to translate this whole process. This picture right here is a little bit technical, but it does show how the ribosome, which can really be envisioned as a very large hamburger bun, where the top is nice and fluffy and the bottom's a little bit shallower, kind of fits on what I call the bacon. If you think about a hamburger and you think of the top of the hamburger and the bottom of the hamburger, think about the fillings that are in the middle, especially a piece of bacon that might lay in there. That is very much how RNA is fed through the two pieces of the ribosome. Think about feeding bacon through a hamburger and you have that envision. Except our RNA isn't really bacon. Instead, it's a copy of that DNA code, and we read it 3 sets at a time. Every 3 RNA bases get translated into 1 amino acid. The result is basically a growing polypeptide chain or a chain of amino acid, and this constitutes the basis for a protein. We know that that polypeptide chain will eventually fold up into a globular protein that is functional. And so from here we see how DNA goes to an intermediate step of making RNA, and then the RNA gets read by the ribosome and translated into something totally new but functional for the cell. Thank you very much, and I'll see you for the next refresher lecture.